Having now covered equilibrium, learning objective number 3 moves on to the receptors for hearing. As a bit of an overview, hearing is the physiological process through which sound waves are detected, transmitted, and interpreted by the brain. It begins when sound waves enter the outer ear and travel through the external auditory canal to the tympanic membrane, causing it to vibrate. These vibrations are then transmitted through the middle ear via the ossicles to the inner ear. In the inner ear, the vibrations cause fluid in the cochlea to move, stimulating hair cells along the cochlear membrane. These hair cells convert the mechanical energy of the sound waves into electrical signals, which are then transmitted via the auditory nerve to the brainstem and ultimately the auditory cortex in the brain. So the input for hearing occurs in the form of sound waves. The characteristics of the sound waves and how they stimulate the fluid and the hair cells in the cochlea allow us to distinguish different sound characteristics. For example, the frequency of a sound wave or how closely the high and low regions are spaced together determine its pitch. The higher the frequency or the closer the waves are together, the higher the pitch. The greater the amplitude or the size of the sound wave, the louder the sound, with sound intensity or loudness measured in decibels. Now, before we take a look at the receptors, a little bit more detail about how the sound waves reach the receptors in the cochlea. And so, to begin, the sound waves are directed into our external auditory canal by the auricle. The sound waves strike our tympanic membrane, and the membrane begins to vibrate back and forth in response to these waves. The middle of the tympanic membrane is attached to our malleus, and the malleus, incus, and stapes pass on these vibrations to the inner ear. The vibrations at the oval window are 20 more times vigorous than those at the tympanic membrane due to the amplifying effect of these 3 ossicles. The movement of the stapes at the oval window creates fluid pressure in the perilymph of the cochlea. We have perilymph in this section here and in this bottom section here. As the oval window bulges forward, it pushes on that perilymph or the fluid, and that movement of the fluid moves all the way around to the round window, which now bulges into the middle ear so that this fluid has somewhere to go. Now, the pressure waves or the movement of the fluid in this top section, which is called the scala vestibuli, causes movement of the vestibular membrane, which is a membrane which runs just along this section here. The movement of the vestibular membrane causes movement of the endolymph inside the cochlear duct. So this is our cochlear duct here, and the movement of this vestibular membrane causes movement of the endolymph or the fluid within that cochlear duct. And then that movement of the endolymph or the fluid inside the cochlear duct causes movement of the basilar membrane, which runs along this section here. Now, our hair cells of the organ of Corti, which is also called the spiral organ and is our specific receptor for hearing, sits along this basilar membrane here. So recapping all of that for you before we go on into any more detail, we have sound come in through the auricle, in through the external auditory canal to our tympanic membrane. That membrane vibrates in response to the sound waves and transmits those vibrations across into our 3 ossicles. These ossicles amplify this vibration. Our stapes, which is our last ossicle, connects to our oval window. It passes these vibrations into the perilymph in these sections along here. As that fluid moves, it must have somewhere to go, so the round window and the membrane covering the round window will bulge a little bit into our middle ear. The movement of the perilymph in this section here will cause movement of the vestibular membrane, which sits along the top of this red section here. The movement of that vestibular membrane causes movement of the endolymph within the cochlear duct. Movement of the endolymph causes movement of this basilar membrane, and extending out of our basilar membrane is our hair cells, which make up the organ of Corti and our receptor for hearing. Now, I am going to recap all of this in text for you, but before I do that, we need to take a bit of a closer look at the cochlea and those 3 channels that run inside the cochlea. And so we have that top channel, which is the scala vestibuli, the bottom channel, which is our scala tympani. Both of these channels contain the perilymph. These channels are actually continuous with one another. Our middle channel is our cochlear duct, and our cochlear duct contains endolymph. So as the sound waves travel through the external ear and the middle ear to that oval window, where the vibrations of the ossicles cause vibrations of the membrane covering the oval window, this causes pressure waves to form in the perilymph, in our scala vestibuli and our scala tympani. The movement of this perilymph in the scala vestibuli, or this top channel, causes movement of the vestibular membrane. So our vestibular membrane sits along here, which in turn causes pressure waves or movement of the endolymph within our cochlear duct. So that's this middle channel here. Movement of the endolymph within our cochlear duct causes movement of our basilar membrane. So our basilar membrane runs along here. Our basilar membrane contains the hair cells, which make up the organ of Corti and is our receptor for hearing. Now, when the basilar membrane moves, it causes hair cells that extend out of that basilar membrane to bend up against our tectoral membrane. So this white part here is our tectoral membrane. Because the hair cells are bending up against this tectoral membrane, they will bend and a receptor potential will be generated. So zooming in once more, now we're going to take a bit of a closer look of this organ of Corti, which is our specific receptor for hearing. And so movement of the endolymph within that cochlear duct, which is the middle channel, causes our basilar membrane to move. Movement of the basilar membrane causes our hair cells, or our stereocilia, to also move. These hair cells then bend up against our tectoral membrane. When these hair cells bend, a receptor potential is generated, and this now runs along the cochlear branch of our vestibulocochlear nerve. So our basilar membrane moves in response to movement of that endolymph. That causes movement of these hair cells. That bends these stereocilia, or the tiny little hair cells, up against our tectoral membrane. That bending causes the receptor potential to be generated. So putting all of that together in a written summary, sound waves are directed into the external auditory canal via the auricle. Sound waves strike the tympanic membrane, causing vibrations. Vibrations transmitted from the tympanic membrane through the ossicles. They amplify by up to 20 times through the ossicles. Vibration of the stapes, which is that last ossicle bone at the oval window, causes movement of perilymph in the scala vestibuli. Movement of perilymph continues to the scala tympani, which now pushes up against the membrane of the round window. Movement of perilymph in the scala vestibuli, so we're now back in that top channel, causes vibration or movement of the vestibular membrane. Vibration of the vestibular membrane causes movement of the endolymph within the cochlear duct, which is that middle channel. Movement of endolymph in the cochlear duct causes vibration of the basilar membrane. Vibration or movement of the basilar membrane bends up the hair cells against the tectoral membrane, and bending of the hair cells generates the receptor potential. Now lastly, once a generator potential is generated from either the hair cells in our semicircular canals or the vestibule or the organ of Corti, they then synapse with the first order sensory neuron, and the combined graded potentials or action potentials will travel along our vestibulocochlear nerve or our cranial nerve number 8. So just wanted to point out that from our semicircular canals and our vestibule, so carrying the sensory information about equilibrium, these impulses travel along the vestibular branch of the vestibulocochlear nerve. So that's this section here, coming from our semicircular canals and our vestibule. From the organ of Corti, the nerve impulses travel along the cochlear branch of the vestibulocochlear nerve, and so that is this section here. Eventually, that nerve combines and then it goes off to the various regions of the brain.